Good morning. Today we will discuss final results that are presented in the paper that we are discussing from the past few days. And in this paper, they have discussed about the expression of sTRAs and CA19 glycans to predict the time to progress of the cancer cells within the patients. So they have the data about 45 patients, and they check the expression of sTRAs and CA19 levels with the corresponding time to progress that they have observed. And what they have find out is that when they check it for this CA19 or sTRAs individually, they this time to progress data does not match up with the actual patient's time to progress data. However, when they check it for the dual labels in the patient samples, they are able to find out that it matches with the time to progress data that they have observed within the patients. So from this, it seems like that both the expressions are important in order to predict time to progress data of the pancreatic cancer cells. And these things they have validated using the panel B, where they have checked these dual expressions of the tumors are mainly separately found in this long time to progress data and short time to progress data. That is, they have dual expressions are distinguished whether the above a particular threshold, dual expressions are more likely to predict time to progress in the long term, whereas below a particular threshold levels, the dual expression data are able to predict the short time to progress of the pancreatic cancer cells. And these things they have validated using the volcano plot also, where they can see that uh, long term uh, long time to progress and short time to progress volcano plots shows that uh, within a particular region, this CA19s, CA19-9 are more expressed, whereas sTRAs are, let me re-explain this figure. So within a particular threshold levels, the short time to progress are more expressed in this CA19-9, and long time to progress are more expressed in sTRAs only. However, these things are common for these high dual tumors. That is, they have uh, tumors that has more expressions of these sTRAs and CA19-9. So within a particular thresholds, we can see that they reflects this long time to progress, and below a thresholds they reflects more towards this uh, short time to progress. That is below 7, approximately below 7% of CA19-9, it will show long time to progress, whereas above 7% of CA19-9 it will show short time to progress. And these things they have validated using this matrix, where they have checked the data, and they can see that up to a particular thresholds, when all the three markers are present, that is dual labels or sTRA19s or CA19-9, they can see this short time to progress in the patients, which is the true positive, whereas some of the samples shows false negative also. And above a particular threshold, we can see that if all the threes are present, that is, all the threes are present, that is dual labeled sTRAs and long-term exposures, long C99 dual labels sTRAs and C99 above particular thresholds, then we can see some of the long time to progress expressions, and these are true negative. These are the case of true negative, and some of the false positive are also shown, where C99 is high but the other things are low, that is sTRAs are low and dual labels are low. So this is the false positive, as it has only one. C99 expressions are higher, but it shows the long time to progress patients, and these things indicate that grouping of the samples are important before doing this analysis, because otherwise it will give otherwise efficiency of this analysis will go down. And then in terms of this H&E staining, we can see that in the short time to progress we can see all the three things, that is dual labels, sTRAs only, and CA19-9 only. And this is usually the more differentiated cells that can be seen the top row figures. And in the case of this other case where the sTRAs are more predominantly expressed, here we see that cells are not differentiated but are segregated. It can be seen in this panel A, which shows the sTRAs expression. The zoom in image shows cells are segregated and our vacuole formations are there. And here the another staining where it shows that the vacuole formations are there and dual expressions and CA19-9 expressions are also visible. However, in the case of long time to progress, we can see that it is also a poorly differentiated and large vacuole formations. At the same time, it has the distinct sTRAs only or CA19-9 only expressions more predominantly expressed. However, in some cases, we also see the dual expressions are also there. That is in the 101 group cases, the dual expressions and CA19-9 expressions are there, but only sTRAs expressions is less visible in this case. So all this data finally indicates that the time to progress can be predicted by grouping the samples, by grouping the patients that has CA19-9, sTRAs and dual labels. All the three are present. Then there is a high possibility that the disease will progress in short duration of time. A short time to progress will occur. So I hope you understood how they come up to this point that presence of all the three conditions, that is sTRAs only, CA19-9 only, and dual expressions will cause those patients will have more possible chances that those patients will have short time to progress of the pancreatic cancer cells. And some of the false positive and false negative chances are also there, that is shown in this panel C. So if you need to understand this bioinformatics of this analysis in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.